Welcome back, students. Today we are going to get to apply what we've been talking about in terms of electronic spectroscopy to the investigation of biomolecules. So this is some of the stuff that I did in grad school, so naturally that makes this one of my favorite lectures. In the past few lectures, we've been looking at basic techniques for electronic spectroscopy. We've discussed how molecules absorb light and make transitions between electronic states and different vibrational states. And one of the key things that's going to be important for describing biomolecules is this idea of chromophores. So we talked about chromophores last time, and remember the key function of chromophores is that they are going to absorb different wavelengths of light than maybe the rest of the molecule. This is especially important for large biomolecules. And they're also relatively independent of the rest of the molecule. So we can figure out what the molecule is doing just by looking at this one chromophore. And this makes chromophores very well suited in the study of large biomolecules. So the learning goals for today are going to be to describe the importance of chromophores like we just discussed. So without these chromophores, we're not going to be able to get a lot of information about large molecules. And then we're going to go through a couple of examples. We're going to talk about fluorescence, specifically this fluorescence technique called FRET. And then we'll also talk about circular dichroism. And both of these turn out to be pretty useful to describing protein folding. So we'll talk about how these techniques can be applied to study protein folding, and there's a lot of other potential implications for these techniques as well. So let's start by just thinking about proteins. We remember proteins from biochemistry, and proteins, as well as other biomolecules like nucleotides and so forth, are big. Now, my background is as studying protein folding. I did a lot of spectroscopy in studying protein folding. So that's where I'm going to be teaching from that angle today, but these techniques could be applied to many things as well. So here's an example of a protein, and this protein has a very large structure. Now, this is much more complicated than anything we have discussed. We've been basically limiting ourselves to triatomic molecules and maybe molecules with 4 atoms if we're feeling spicy, but this molecule has over 2,000 carbon atoms, let alone hydrogens, oxygens, nitrogens, and sulfurs because it's a protein. So proteins are also complicated because not only do they have a whole lot of molecules or atoms in them, but when they have a whole lot of atoms, what comes with that is different types of structure. So we know we learned this in biochem, that's primary, secondary, tertiary structure, potentially quaternary structure. And they're also not static. We've been talking about vibrational modes and so forth. So these bonds vibrate, they rotate, and these proteins are fluid molecules. So all of this makes doing spectroscopy challenging. So we're going to discuss some techniques that can actually be used despite all of these complexities in folding, and they're all going to rely around chromophores. So ignoring most of what the molecule is doing and only looking at certain aspects. So the first biologically relevant chromophore I want to mention is going to be a natural one. So the natural chromophore, as you might remember, is tryptophan. So tryptophan, I don't have a structure of it on this slide. We can draw it up and look it up in class if we want. Tryptophan is a big planar side chain. It's nonpolar, and it's aromatic. Tyrosine is aromatic as well. And both of these absorb light significantly at 280 nanometers. And it turns out that cysteine also absorbs a little bit of light at 280 nanometers due to its sulfhydryl group there. So with these 3 amino acids, we can study them and figure out how much light they absorb. And so that's what these numbers are here. This comes from a paper from '89 from Gillen von Hippel, where they studied the absorption of light based on these chromophores here. And they have these values here. So we can see that tryptophan absorbs 5,690 nanometers of light. So these are the molar extinction coefficient using Beer's law. So Beer's law tells us that the amount of light that a molecule can absorb is equal to this, what I have here is epsilon Cl. Epsilon is this molar extinction coefficient. That's what we're showing here. And C is the concentration and L is the path length of the instrument, which is often 0.1 centimeters. So what this tells us is that these different chromophores can absorb different amounts of light, and these are molar extinction coefficients. So what this tells us is that 1 mole of the tryptophan side chain can absorb 5,690 amount of light per molar per centimeter there of the path length. So if we have a protein and we know how many proteins, tyrosines, and cysteines our protein has, which we do because we basically have all the information about every protein in most every organism, and new organisms are being sequenced all the time. We can just check out the sequence of any protein we want, figure out how many tryptophans, tyrosines, and cysteines it has because we sequence the genomes. Then we can add these together and actually figure out what our protein would do. So if our protein has 2 tryptophans, 10 tyrosines, and 1 cysteine, 1 mole of our protein would have 2 moles of tryptophan, 10 moles of tyrosine, 1 mole of cysteine, and we can have a molar extinction coefficient for our protein. And this is what we need to determine the concentration of our protein. So the L, the path length, is standard based on the instrument. So if we know the molar extinction coefficient, we can just measure the absorbance of our protein at 280 nanometers if it's pure and figure out its concentration. So this is one very powerful way that we can use to figure out the concentration of a protein using spectroscopy. Another biologically important technique is fluorescence. So last time we talked about fluorescence and how fluorescence works. We saw an example of labeling the nucleotides with fluorescent tags to allow us to detect which nucleotide is being synthesized. So we already talked about that. One other really important example is green fluorescent protein, GFP. So I actually did a lot of studying on GFP-like proteins in grad school, so I'm pretty familiar with them. Here we have our GFP crystal structure. You can see the chromophore is inside. GFP forms an 11-stranded beta barrel, which is pretty interesting. It's non-sequential in any way. Its folding is actually pretty complicated. And it auto-catalyzes its own chromophore. So I have this mechanism here. And this is a proposed mechanism. The exact order of these steps isn't really known, but this is the chromophore that's formed in these fluorescent GFP molecules. And you can see that it is an extended pi system. We have our tyrosine here, and then we have a double bond that gets introduced with this dehydration, and we have a cyclization reaction as well. So we have this pretty strange amino acid, I guess. And the chromophore here formation actually is in the peptide backbone. So we get this new weird thing, and it's this big long pi system, and this big long pi system is fluorescent. So it can absorb some wavelengths of light, and then it emits green light, and we have this green fluorescent protein. Now, why is green fluorescent protein so useful? The main reason that it's useful is that we can basically encode it onto anything. So we can take any protein that we want, and we can put it on a plasmid if we're working with bacteria. There's more advanced techniques for working with mammalian cells and so forth. But we can simply take any protein of interest that we want, go into its open reading frame, and attach the sequence for green fluorescent protein to the open reading frame of any protein. What do we get then is we get a protein that has green fluorescent protein attached to it. So we actually can introduce fluorescence inside of living cells without having to tamper with the living cells in any way. We don't have to try to do chemistry inside of a cell to label molecules. So that makes GFP a very powerful technique, or I guess employing GFP is a powerful technique for doing imaging inside of cells. So you can imagine, and experiments have been done to do things like tag proteins that go to the mitochondrial membrane, and then you can see what the mitochondria are, or the nuclear membrane, or the exterior membrane. You can just see, where is my protein? Is my protein cytosolic? Is it nuclear? Does it get secreted? And if it's fluorescent, you can track where your protein is going. One thing, or one downside for green fluorescent protein and you might be able to think about this as well, is it's kind of big. So it's a protein, it's about 200 amino acids. So to attach an about 200 amino acid protein to another probably about 200 amino acid protein could affect what it does. And so that's something to keep in mind with this kind of fluorescent tag, is that is in fact a very large fluorescent tag. And there's assumptions often, and sometimes they're explicitly controlled for in really good studies, that show that it doesn't seem to affect the function of the protein. That's always something to worry about. When fluorescent molecules absorb light, they are excited, right? And we saw this pathway last time. Let me go back to it here, just so we can look at it. So we have this state and our proteins are being excited. So a green fluorescent protein can be excited. It gets into this excited singlet state, and then it descends down to this ground state. And we talked about how long it does this. There's a fluorescence lifetime, and we call that tau, and that talks about kind of how long it's in this excited state. And we'll talk more about this explicitly next semester. That has to do with chemical kinetics. But this tau corresponds to the amount of time it takes for 2/3 of the molecules to fluoresce. So after one tau period of time, only 1/3 of the molecule is still in the excited state. Now, it turns out that fluorescent molecules have other ways that they can get back down to the ground state without fluorescing. So we talked about this a little bit. They could be internal conversions, and those are kind of boring. But it turns out that there are other types of conversions that can happen that lead to different wavelengths of light coming out. Now, what do I mean by that? Let's go back to the slide we were on. And another thing that our proteins can do is called FRET. So FRET here stands for fluorescent resonance energy transfer. In some cases, you might see this referred to as Forster resonance energy transfer. Forster was the guy who first researched this, so there's a little bit of confusion on what that F should actually stand for. It's called both. Now, what happens in FRET is that we have a molecule. So we have two different fluorescent molecules. One is the donor, one is the acceptor. The donor fluorescent molecule absorbs light, just like it always does in fluorescence. It loses a little bit of energy. Then instead of emitting various wavelengths of light, and we see these broad peaks, of course, because we have different vibrational and rotational states that can go on. Instead of emitting that light, what it does is it passes it on to a second chromophore. So here we can see that our donor emission wavelength overlaps with the absorption wavelength of a second chromophore. So this process of passing the energy on is the first chromophore's way of removing the energy through a non-radiative pathway. So there's no light emitted, it just passes on its energy to a second chromophore. So that second chromophore is now in an excited state, and then it emits light. And so what happens here is, in a lot of cases, and the good cases of this, is that we have absorption. So if we're absorbing light here, so say we use this wavelength, only our donor chromophore can be excited. So the absorption of our acceptor is not excited at this wavelength. So we only excite the donor, and then if we measure acceptor fluorescence, it's telling us about how this FRET process has occurred. Now, you may imagine that this process is extremely distance sensitive. So if our chromophores are too far away, they're not going to be able to transfer energy to each other. They have to be close. So not only does this technique kind of is a way that the molecules can remove energy, but it also gives us ways to actually measure the distance between molecules. So the way that this is normally done, and we'll see an example of this on the next slide, is that fluorescent dyes are attached to something like a protein that are two different regions that are far apart if the protein is, say, unfolded. And then when the protein folds, you now have these two chromophores brought close together, and so we will have FRET during the folded state and no FRET during the unfolded state. This could also be done to do things like talk about binding. If you had two dimers or two monomers that form a dimer, or any sort of other binding event, you could do this kind of thing. And really, a lot of work has gone into evaluating this technique here. So I have an example of my own from my research I did when I was in grad school. Now, unfortunately, we actually didn't want FRET to happen, and FRET kind of messed us up when it did happen. But I worked with some fluorescent proteins, and you can see here. So I have these are they're called KB and YK, and I'm happy to talk about these in class if you guys are a little bit interested. But we have the excitation wavelength for KB. We have the emission, and you can see that this emission wavelength heavily overlaps with the excitation wavelength of our yellow fluorescent protein here. So we can excite at the cyan protein and measure the emission of the yellow protein. This, of course, like the other technique, also has some caveats. So the main caveat from this is that these fluorescent dyes that are often attached to proteins. So in this case, these are like green fluorescent protein like things. So this doesn't come into play for here. But sometimes dyes are attached to proteins and they want to study, say, how protein fold. But when you attach these big dyes that are often large and nonpolar, you could potentially be affecting how these proteins are folding. If you give them a whole lot more hydrophobic things, maybe the hydrophobic collapse get overweight or so forth. Another thing to note is that not only is FRET distance dependent, but it's also angle dependent. So depending on the angles between the two chromophores, they can kind of get messed up. And that is usually not dealt with. It's usually averaged over and say, well, on average, we expect the angles to line up good this amount of time. And so we can just slap a constant in there to talk about that. So that's normally how that gets handled. Now let's go ahead and look at this experiment here. What we have is a polyproline chain. So you can see there's a whole bunch of prolines here. And to either end of the polyproline chain are attached these dyes. So here you can see a little bit about what I mean when I talk about these dyes are being big and hydrophobic. So what happens, and what this is showing, is different lengths of polyproline chains. So 6 prolines in a row, 11 in a row, 14 in a row, up to 40 in a row. And when you have very small amounts of proline, the two dyes at the ends are very close together. And so what we're plotting here is the FRET efficiency, or the amount of the acceptor fluorescence that we measure compared to what we expect. And that is very high. This is very efficient because they're very close together. As the molecule gets longer, we start to see a population that is unfolded, that is far apart. And this gets more and more and more of the case the longer the protein gets. So in this case, it's just simply looking at a marker, sort of a baseline for this technique. But you could imagine monitoring protein folding with this and looking at how these two dyes get close together, say as a function of time or guanidinium concentration or temperature or so forth, to talk about actually making measurements of how proteins are folding. Another very useful spectroscopic technique is called a circular dichroism, or CD. And so this technique uses circularly polarized light. So that's going to be this one here in the middle. And what happens when molecules absorb circularly polarized light, it depends on the chirality of the molecule. So maybe you've seen this in organic chemistry lab looking at different chiral molecules. But biomolecules are all chiral. So all of the amino acids, except for glycine, are chiral, and they all have the same chirality. They all form things like helices and sheets with certain chiralities because they're all made up of the same chirality. And this allows these molecules to be active under circularly polarized light. So what circular dichroism can do is it can distinguish between different types of specifically secondary structure, because these things like alpha helices will interact with circularly polarized light in a different way. And so here in this plot, we can see the red curve, which is a plot of what happens when you have only alpha helices in the CD, how alpha helices absorb light. You see in green how beta sheets absorb light. And then beta turns as well, pretty similar. And then in purple here, we have unordered or unfolded protein, that the way that absorbs light. Now, the book, if you read this section in the book is going to tell you that you can use this to figure out how much of your protein is alpha and how much of your protein is beta. That is an oversimplification of how this technique actually works. So if we look at alpha and beta structures here, we can see that their peaks are kind of in the same spot. And that makes it actually really hard to deconvolve them and figure out what is what. So if we go back to our GFP slide here, I ran the CD spectrum of GFP in one of these programs to try to figure out what percent was alpha and what was beta. And it told me it was about 50-50. And you can see here that it's almost exclusively beta. So there's a little bit of alpha helix here. There's some of these curves that you could describe as like one turn of an alpha helix, but it's almost exclusively beta in the program, which is totally incapable of predicting that. So does that mean CD is useless? No, of course it doesn't mean CD is useless. How do we use it? So you can see here, this is some of my actual data from grad school. And these are the kind of peaks that I got. And you can see that there's basically no way you're going to be able to pull this apart and figure out how much of this is alpha and how much of this is beta. But what we can do is look at this and figure out what's happening, say, as a function of temperature. So alpha and beta kind of have peaks in the same spot, but they're both very different from this disordered in purple here. So what you can do is measure the CD signal of a protein as a function of, in this case, what I did was temperature. And you can look at that. And what we have for this protein is it stayed folded, which means it had this sort of signal over this temperature range. So at 20 degrees, 40 degrees, 60 degrees, it all looked like this. Then once we hit 80 degrees and the actual melting temperature is, I think, about 79 degrees Celsius, we started to convert to unfolded. And after that, above that temperature, it looked much more like this unordered. So the CD signal changed from being down here to being up here as a function of temperature. And so I was able to monitor protein unfolding as a function of temperature. CD is also very useful for saying I've made some change to my protein and it hasn't changed the structure. That's often what it's used for. So you can't predict how much alpha and beta a structure is, but you can do something like add a big old fluorescent dye, tag to it, measure the CD and say, oh, it looks the same. That means my secondary structure is the same. Or you can do mutations or so forth, or add a his tag and look at the CD structure. And then you can be pretty confident that whatever change you made to the protein is not changing the structure of it because the CD says it has the same type of secondary structure. I have a few more slides that we can go over in class. There's some other interesting things you can do, other things that I've done in grad school. And I'll mention a little bit in class about single molecule spectroscopy. But for now, I am going to let you go. And I will talk about this basically the whole class period. I really do like this stuff. It's really fun, I think, to apply these techniques that we're learning here in physical chemistry to biological systems. So to conclude here, we have electronic excitations that are requiring ultraviolet and in some cases visible light to excite. When we excite these electronic transitions, we also pick up vibrational and rotational transitions just like we've seen before. And we have a different selection rule. In this case, we use the Frank-Condon factor, which talks about the overlap between the vibrational ground state and whatever state it's going to be excited to. The more overlap we have, the stronger the transition. Chromophores are a very nice way to get meaningful information about larger molecules, whether that molecule is, one of our conjugated polyenes or green fluorescent protein. We also saw electronic selection rules, and these selection rules are even less stringent than our other not very stringent selection rules, but we still have a dominant pathway based on these selection rules. And when a molecule is in an excited state, it's pretty complicated what could happen to it. It could go through non-radiative pathways and go back to radiative pathways, but it's not as simple as absorbing and emitting a photon of the same intensity. And we've used some of these techniques to look at biomolecules. So with that, I will let you go, and I'm looking forward to talking with you guys about this in class. And from here, we're moving into a very different topic next time that's going to be about group theory. So I'm looking forward to talking with you about that as well.